Hey Laura, good morning.
Hi, Sam. How are you?
I'm doing okay, thanks. How are you?
Good. You know, just look at closing in on us, this little virus, huh?
I know.
Do you have anybody sick in your building?
Not that I know of in my building. I think we've been okay so far, but my sister is sick.
No.
Yeah, she's in North Carolina. Um, but she's okay. I mean, she's fine. She's young, she's healthy, she's fine. But it is kind of scary when it hits home like that, you know?
Absolutely. Well, I hope she'll be okay. I mean, it's just, yeah, there's been some, I guess, incidences around campus. So,
So we do not forget to do that. And we are recording. And we're going to go ahead and admit everybody. All right, welcome everyone. I see you're all coming in right now. We have a very interesting webinar today. We're really excited to see you all here. I'm always doing a bunch of admin stuff as I'm talking, so I'm sorry that I pause. And this video is being recorded, so you can see it on our website and you can see it on our Facebook page as well. And it's lsuhealthfoundation.org. So you'll be able to see that there. Just give us a couple hours to get it up after the webinar ends. And I'm going to share a PowerPoint for y'all. One moment. One second, sorry. Okay, can you see the PowerPoint, Laura?
Can see it.
You can? Okay.
yep.
Great. And so this is for CME credit, just to let you all know, you can get CME credit if you are at LSU. Please, if you can, change your email address to your LSU email address, and that'll make it a lot easier for Miss Bell to find you. Take that off, and I'm going to end screen sharing. And I'm going to turn it over to Crystal Oliveira. She's our Chief Development Officer at LSU Health Foundation.
Thanks, Sam. Welcome, everybody, to our ongoing coronavirus webinar series. Today, we have a special webinar. It's an update from a previous webinar we did a couple of weeks ago in the very early discovery stages of a really exciting project that's coming together with two physicians. One is at Tulane, Dr. Mari Nover. She is the professor in the Department of Microbiology and Immunology at Tulane University School of Medicine. And then our own Dr. Paul Fidel, and he is our professor and head of the Department of Oral and Craniofacial Biology. He's also the director of the Center of Excellence in Oral and Craniofacial Biology and the Associate Dean for Research at LSU House School of Dentistry. Welcome to both of you. We're excited about this topic, this MMR, and what the potential benefits can be for us. Take it away.
Great. I will start by screen sharing.
And I'll just do a short introduction, just let you all know. Thank you for joining us. Mari and I have worked together for a very long time, and we've worked on several models of candidiasis. And we had our labs together for a long time, and that worked really well because we're married. And about a year and a half ago, she had a great opportunity at Tulane, so she sort of jumped ship, but in a very good way. And so we've been still working very closely, and a lot of the work that we've been doing sort of spawned this whole hypothesis and concept. And it's actually very timely because with all the upticks of cases now and the just tremendous increases, it would be really nice to have an action that would sort of sequester things and soften the blow a bit. So Mari was actually the brainchild behind putting all the pieces of the puzzle together to identify and propose this concept. So I'm going to let her take it away with the start of those thought processes, and then I'll pick it up later.
Okay, thanks, Paul. So I'm gonna forward here. All right, so this all started back in early March when I began poring over a lot of the data that was coming out of China with their early outbreak. And this is this is about the time when the outbreak was actually declared a pandemic by the World Health Organization. So I started looking at all of the data and kind of putting these clues together in a thought mapping exercise. So some of these clues that I'll go over in more detail in the subsequent slides. First of all, they saw no mortality in children under 9 in China in the outbreak. The mortality was primarily due to sepsis. And we also know that live attenuated vaccines have the potential to dampen sepsis, and that is work that's from our laboratories. Children we know receive several live vaccines throughout childhood, and they therefore have more recent live vaccine exposure. So the thought was, could this live vaccination exposure protect against the sepsis that's associated with COVID-19? So that was the gist of what I came up with, and I tweeted that out back on March 15th with all of the details and the supporting data. So the first clue was very low mortality in children. So very low detection rates were observed and no reported deaths in children under the age of 9 with SARS-CoV-2. So no fatalities. Again, this is the early data out of China. This was published by on February 28th by the World Health Organization. Importantly, I also found in looking back at other coronavirus outbreaks that cases of infections in children with the outbreaks of SARS-CoV-1 in 2003 and the MERS-CoV in 2012 were also characterized by low rates of infection, mild symptoms, and really good prognosis in children, which was kind of interesting. So there's a pattern here with the coronavirus outbreaks. This is very different than what we see with seasonal flu mortality. So there's really high risk for seasonal flu to affect children. Every year, millions of children get sick, thousands end up hospitalized, and between 100 and 600 children die every single year. And that's why we have a very active flu vaccine campaign that we try to educate parents about the risks and have your children get vaccinated. So the risks are actually higher the younger the child is. So children younger than 5 years old, especially those that are younger than 2, are at really high risk of developing serious complications from flu. So I thought this was strange. So why is there low mortality in children with SARS and MERS, the coronavirus outbreaks, but not with seasonal flu, which is caused by the influenza virus? So two different, very different species of viruses. So I wondered whether this was due to the differences in the disease pathogenesis and the actual causes of mortality in coronavirus versus the flu. So with COVID-19, again, this is early data coming out of China. The earliest reported biomarkers that were associated with patients developing severe disease were identical to those biomarkers we see in sepsis patients. And this really struck me as I work on sepsis in my laboratory. So the COVID-19 outbreak in Seattle, that was the early outbreak in the US, they saw that septic shock was in 70% of these severe patients. So really high prevalence of these sepsis markers. This is different from seasonal flu. So mortality with flu is often a result of secondary infections, including other viral species and bacterial pneumonia in the lung, and exacerbations of underlying chronic conditions. So you often see mortality with people with underlying health problems, especially in children. So I've been talking a bit about sepsis. What is sepsis? This is just a term that's for life-threatening organ dysfunction that's caused by a dysregulated hyper-inflammatory host response. So basically your systemic immune system goes absolutely haywire and a lab elicits this massive inflammatory response that leads to organ damage. It can be caused by systemic, It can be caused by systemic bacterial, fungal, or viral, or polymicrobial infections, but these are often systemic. They can originate from various parts of the body. They can originate from a lung infection, from an abdominal infection, or bloodstream infections. The diagnostic criteria for sepsis is a high SOFA score. So that is a sequential organ failure assessment score. We saw high SOFA scores in the COVID patients, fever or hypothermia, also impaired breathing and blood oxygen level. So biomarkers, things that we can actually measure in these patients, would include things like the cytokine storm, so massive upregulation of inflammatory cytokines, and also markers of coagulopathy, so dysregulation of coagulation. You see blood clotting problems in these COVID patients. So D-dimers is one of the major biomarkers for that. And I also use these same biomarkers in my sepsis model in my lab. So how do we get from there in linking this to potentially protection from early childhood vaccination? So I wondered whether the lack of mortality in children with COVID-19 could be due to more relatively recent exposure to live attenuated vaccines. So we know that live vaccines in particular are associated with generating beneficial non-specific effects, these NSE, against completely unrelated infections. This is also called heterologous immunity as opposed to homologous immunity. And there are documented non-specific beneficial effects from live vaccines such as MMR, polio, and BCG, and I'll be getting into those in just a bit. We have a considerable amount of supportive epidemiological data for this. So what do I mean by a non-specific effect? This would include things like reduced hospitalizations and mortality to other common infant and childhood infections, and these are often respiratory. So there's a really, if you want to read more about this, there's a series of excellent review articles that were published in Clinical Microbiology and Infection that is listed below here if you want to look those up. So in terms of live attenuated vaccines that have these NSE, these non-specific beneficial effects, first and foremost is BCG. This is the bacterial tuberculosis vaccine. It is Mycobacterium bovis BCG strain. It's a related strain to TB. It's about 99.9% similar to TB at the genetic level. There are current national BCG vaccine campaigns in many, many countries worldwide, but the US is not one of those, as you'll see here in green. Some countries in Europe had past national BCG vaccine campaigns, but no longer use it, but had been used historically. And I'll get into why that is. So why don't we use BCG vaccine in the US at all? And first and foremost, TB is not an endemic infection in the US. We don't have very high rates of TB unless you're looking at an urban environment such as New York City. Also, the BCG vaccine doesn't really work that well against protecting against pulmonary tuberculosis. So it's only about 50% effective, so low efficacy. It's also a subdermal vaccine that causes a lesion and a scar similar to the smallpox virus. So you have to inoculate subdermally and you get this lesion that scabs over. So the fact that it leads to scarring will limit the ability to do blinded studies with clinical trials. The subjects will know whether they've received the real vaccine versus a placebo because the placebo will not scar. It also leads to an adaptive response and seroconversion against tuberculosis that limits the use of the PPD diagnostic test. That's the test that we use for detecting tuberculosis infection, you'll see here on this image. So the PPD test relies on a cell-mediated response to TB. It's just a delayed type hypersensitivity. And the BCG vaccine will actually cause this. So you're no longer able to use PPD to tell whether someone has active TB, and you have to use other diagnostic criteria like chest x-ray. So for all of these reasons, that's why the US doesn't use BCG anymore, or doesn't use it at all. There are other live attenuated vaccines that have documented non-specific effects. Many of these are viral vaccines. So smallpox vaccine, but that was discontinued in 1972. Oral polio vaccine, also discontinued in 2000. Now we only have an inactivated vaccine available. The shingles vaccine, which is used in older adults, they actually replaced that with a more effective non-live vaccine in 2018. So that's no longer used. the only one that we have that's currently used readily in the US is the measles vaccine, which is just the measles vaccine in low-income countries, or MMR in high-income countries. That would cover measles, mumps, and rubella, so 3 live attenuated organisms. Also, the chickenpox vaccine is a live attenuated vaccine, but much less data on that with these non-specific effects. Okay, so we've known about these non-specific effects for quite a while, going back as far as the 1800s when we introduced the smallpox vaccine. There were also already reports of reduced non-specific morbidity in children, leading up through the 20s when BCG was introduced, as well as in the 60s when we had the polio vaccine introduced, all the way up to the time point when we started vaccinating against measles virus, and now more currently with MMR. So this was probably in the last 20 years or so is when we became aware that the mechanism of action of these non-specific effects was actually due to training the innate immune system rather than the typical way a vaccine works, which is to induce an adaptive response. Okay, so what do I mean by trained innate immunity? This is a relatively new concept, a new term in terms of immunology. It just means that you have a non-specific memory immunity that's mediated by these innate cells. So not a T or a B cell. These could include macrophages and monocytes, which were the cells they first discovered had these trained non-specific activity. So the innate cells actually become trained by interacting with live microbes. So that's why it's so important that the vaccine be a live attenuated vaccine. Does not work when you have these cells interact with killed microbes. So the mechanism is that you get epigenetic reprogramming of these innate cells after they've seen the live attenuated vaccine. So you get reprogramming at the epigenetic level that leads to enhanced responsiveness to a secondary infection. So the second time you're exposed to an unrelated pathogen, you can get an enhanced response. So how do these live attenuated vaccines actually end up training the innate cells? And most of what we know today comes from work with BCG, which is the tuberculosis vaccine. So we know that BCG as a live vaccine actually traffics to the bone marrow in patients and induces myelopoiesis of stem cells. At this point, it can actually alter the epigenetic profile of these hemopoietic stem cells so that when you isolate differentiated macrophages from bone marrow, they have enhanced inflammatory and antibacterial activity when you work with them in a tissue culture dish. Okay, so the fact that all of the concepts related to trained innate immunity really focused on this enhanced inflammation, that's kind of where I came to a stop point. I didn't understand how this could make sense in terms of protecting against sepsis. So how would enhanced inflammation and trained macrophages help protect against a viral infection that kills by inducing systemic inflammation such as COVID? So we came up with a novel concept from work in our laboratories based on data from our mirroring sepsis model that suggested that live vaccines induce these GR1 positive myeloid-derived suppressor cells, MDSCs, that we know can limit septic inflammation. So we came up with a new concept called trained tolerogenic immunity to explain how a live vaccine could interact in the bone marrow and induce these suppressor cells that can actually suppress septic inflammation. So I'm just going to give you just a brief few slides of data. This is work from our laboratory. So in our hands, our vaccine strain is a live attenuated Candida species called Candida dubliniensis. And our sepsis model is a polymicrobial abdominal sepsis model where we inoculate the animals with Candida albicans and Staph aureus. And this polymicrobial sepsis, you'll see in the red line here, this is lethal challenge only. This is days post-inoculation. They get very sick very quickly, and you have rapid morbidity and mortality over 24 to 48 hours, up to almost 100% mortality. However, when we vaccinate with Candida dubliniensis ahead of time, you can see that these animals do very well. They don't look sick, and we have about 90% protection against the sepsis. Down here, you'll see the sepsis score in our lethal challenge animals. You'll see a very, very high sepsis score very early on, but these vaccinated animals are really protected against sepsis. Now, going back to the idea that they're inducing suppressor cells that lead to protection, we depleted the suppressor cells with GR1 antibody, and you can see those animals succumb rapidly to the lethal challenge. So we know that the GR1 cells are what's responsible for protecting these animals. As an aside, we also depleted macrophages in this study, and they had no effect. So macrophages are not involved in the trained response to sepsis. Okay, so we came up with a completely novel model for innate training called trained tolerogenic immunity, which takes together two of the different previous models for training. So there's trained innate immunity, where you have enhanced responsiveness to a secondary infection. And there's also something known as endotoxin tolerance, where you become very, very tolerant to that toxin. That's another form of innate training. Trained tolerogenic immunity takes advantage of both of these different models so that you get a tempered, more productive response that includes suppressing damaging septic inflammation. So we hypothesized that live attenuated vaccines could be used as inducers of trained tolerogenic immunity and inducing these MDSC suppressor cells to inhibit sepsis. So in discussions over the months after I first tweeted out in mid-March, and discussions with Paul, we decided to put pen to paper and develop the thought mapping exercise into an actual publication. So we're finally able to get this published recently, and InBio was published June 19th. And I will let Paul take it over from here. Are you able to screen share, Paul?
I am trying.
Okay.
Hold on. Yes.
Okay.
All right, I hope everybody can see that. So yeah, we put pen to paper on this and looked at proposed the concept of an unrelated live attenuated vaccine to serve as a preventive measure against the septic inflammation. And we chose MMR for this, largely at the beginning because we thought it was 3 for 1. You got 3 viruses, 3 attempts to try to get these cells produced into the bone marrow and move into the MDSCs to inhibit sepsis. And so in general, the MMR is in its second phase. It's a live attenuated against measles, mumps, and rubella. It is indicated really for the prevention of measles and mumps and rubella in individuals or children at 12 months of age and older. It's usually a 2 to 3 course type of regimen of boosting with the vaccines over about 10 or 11 years. And it's very safe. It has very few contraindications. In general, this vaccine has been around for over 40 years now, and it has really protected many, many children over that 40-year span in a very effective way. There are a few contraindications to getting it as an adult. If you have some type of immunocompromised condition, if a woman is pregnant, if anybody has allergies to vaccines, or a moderate to severe fever at the particular time you're getting the vaccine, it's not a good idea. But otherwise, it's really very safe, and it's an intramuscular administration, and it does not leave a lesion. There's a good bit of mounting evidence now to support the efficacy in general towards this non-specific effects that Mario was talking about. A couple examples are in military personnel. And as you know, the military have to get a series of immunizations during enlistment, and even booster shots, and at least verification of these when they get on, when sailors get on ships for the Navy. But There was an incident on the USS Roosevelt not long ago where almost 1,000 sailors came down with COVID-19, but there was only one hospitalization, and all those sailors had to have evidence of the recent MMR vaccination or have one before they entered the ship. So that's nice supportive evidence. There's also the USS Nimitz that was out at sea in the Pacific and received an MMR due to some exposure to mumps during some military exercises. And they came back and docked in Seattle, really close to where the COVID outbreak seemed to occur in the state of Washington there. And on that ship, as they docked and became integrated with the population, there were no reported cases of COVID-19. This is a study by Jeff Gold, who looked at a lot of epidemiological data from several countries and looking at age ranges of people that were getting more of the infection rate and the sequelae from COVID-19. You can see here that from 50 years in age and up, the death rate got increased substantially. And those that were less than 50 had a much lower rate. And those who were less than 50 years old were likely the ones that had an MMR vaccination when they were a child. Those that were 50 and over probably did not. Now, obviously, as age increases, that potentiates more of the morbidity associated with this, but it's a pretty telling type of statistic between the green and the red. And this is looking at 4 countries: South Korea, Spain, China, and Italy. There's also some very interesting mounting evidence from refugee populations. Usually in cases of refugee camps and so forth, the people entering have to either get the MMR and other vaccines or have had to have them. And in Rohingya, in the refugee camp where they had all received an MMR, they had very low rates of these infections. And that's in Bangladesh there, where they had actually very high rates of infection in Bangladesh overall. Also anecdotal reports from other refugee camps, including those in the United States. When refugees came into the United States, they were required to have MMR vaccination, and really very few cases or deaths associated with COVID-19. Some interesting recent mandatory MMR vaccination campaigns. In addition, in Madagascar, they vaccinated 30% of the population with a measles rubella combined vaccine in 2019, and they had no reported deaths from COVID as late as May of this year. Samoa had a major measles outbreak in 2019 that originated from a traveler from New Zealand. So they instituted a mandatory MMR vaccination campaign, and to date have had no related COVID-19 related deaths. New Zealand turned around and also instituted a mandatory MMR vaccination in 2019 because of that incident in both adults and children. And currently they only have 1,400 COVID cases and only 20 deaths. There's been a lot of personal testimonials as well. Just from our publication, people have contacted us and emailed us certain cases of people getting an MMR for any number of reasons, told us that they came down with COVID-19, tested positive, but were relatively asymptomatic, at least through this period of time. So there's some very interesting circumstantial and anecdotal data, but very much supporting what we're proposing. And you know, in addition, if you look at the efficacy of targeting the host versus the SARS-CoV-2 in the hospital setting and what's going on right now during the pandemic, we see that there's been very low efficacy and actually substantial toxicity of hydroxychloroquine. That's an anti-malarial drug. Had some suspected antiviral activity, didn't do very well. Some modest efficacy with remdesivir, the antiviral that actually is coming into New Orleans in high quantities right now, that does seem to have an effect in late stages, but it's been modest overall. Hasn't been the magic bullet by any means. And convalescent plasma is also being used, and those are anti-COVID antibodies that are donated from people that have previously had the infection. They are showing some effect and efficacy, but it is modest, and it's only modest because the virus has to be outside the cells when it sees, for it to be able to seen by the antibodies to get rid of it. And most viruses like to be inside cells, so it's a catch as catch can type of treatment. But the overall highest efficacy so far, and it is becoming more and more popular now, is use of dexamethasone. And that's basically a corticosteroid. It's an immunosuppressant and anti-inflammatory. But it would be nice if you had something there ready and waiting, as opposed to using that as a treatment under conditions where the symptoms are quite severe. There's also some interesting mounting evidence now that we might have an additional added effect of the MMR, and that is a few mapping exercises from the molecular side that's showing some homologous protein domains in SARS-CoV-2 and with measles, mumps, and rubella viruses. So there's preliminary evidence that the MMR vaccine might provide some cross-reactive protection against COVID-19. It's probably partial, but those antibodies that are produced might actually be able to have an antiviral effect if that cross-reactivity is true. But really, what do we need? This is all very good circumstantial and anecdotal evidence, a lot of potential, but what we really need is the science. And the clinical trials are really necessary with MMR versus placebo in the high-risk population for COVID-19. Also animal models of COVID-19 where you can pre-vaccinate using MMR versus placebo and study and look at the various cell populations as well as the natural history of the infection. But because it's so low risk and sort of a means by which the contraindications are so low, we feel that we can disseminate this hypothesis and related information to the public as really a low risk, high reward potential for adults at high risk for exposure and infection with COVID-19. And by that, I mean healthcare workers, first responders, nursing home residents that are healthy enough to get the vaccine, even sports teams now. We're trying to get sports all the way back. Sports teams are coming back, practices, people are together. It's going to happen, but if we could reduce the sequelae and reduce the high symptomatic nature of the infection, it would be very helpful. It's really no harm, no foul action for adults, with the exception of a few contraindications. So if you really look at the potential effects of the MMR, you have the induction, potential induction of the MDSCs that Mari spoke of to inhibit the COVID-associated sepsis, plus this potential cross-reactivity of the MMR antibodies to COVID-19. Together, if that is really true, that could be a magic bullet against COVID-19. And then you add on that the immune protection against measles, mumps, and rubella, and it's really a win-win. Whoops. And so what are now the next steps and what is happening? Well, I'm happy to say that we are going to be able to start some real science here in the next month or so. We have a newly funded MMR vaccine studies to test this concept. We'll be doing a clinical trial in healthcare workers in New Orleans. This was funded by the Parsons Foundation out of San Francisco, where we will be testing MMR versus a placebo. Those two arms are looking at a primary outcome of the MDSCs in the blood post-vaccination out to a period of 60 days and up to a period of a year, with secondary outcome measures being the health status plus or minus COVID-19 infection if and when it occurs. Again, this is not meant to stop the infection. It is just meant to induce the cells that would be there to protect against sepsis if infected. So we hope that we will be able to show that and help any individuals that would come down with COVID-19 under those high-risk situations. And Mari also has a grant that she got from the FAST Grant COVID-19 program to do an actual non-human primate model of COVID-19 and doing the MMR versus BCG in this case versus placebo. And that will look at the evaluation of those MDSCs post-vaccination as well as the natural history and study that. Mari, you might want to say a few more words about that and then continue from there to the end.
Can you forward to the next slide? I'll roll it out here. Okay, so yeah, we're currently underway with the Primate Center at Tulane to initiate the non-human primate studies with MMR. So that will really give us a good indication of whether or not MMR can actually induce these MDSCs to protect against sepsis related to COVID, and those studies are underway. Hopefully getting additional funding to move beyond the pilot study stage. So over the past few months when we've gotten the word out about these studies, and since our publication came out in MBio, we've been getting a lot of feedback. Most of it is positive, but have to recognize that we live in a world with some mistrust and misinformation about vaccines that has led to a lot of vaccine wary individuals. So how do we change the narrative on vaccines to get people to understand their utility, not just against the actual infection you're trying to protect against, but also these non-specific effects? So first thing I thought of was just re-educating the population on these non-specific benefits of live attenuated vaccines against COVID. So we're trying to market these as a sepsis preventative in this case, as opposed to just calling it a vaccine. And my other thought was to rebrand vaccines and conceptually think about them in a different way. So live vaccines can really be thought of as a novel type of systemic probiotics. Probiotics are very well accepted through oral delivery, and that it's well known that they induce all kinds of beneficial effects on the immune system as well as digestion. Well, let's take this a step further and understand that humans are constantly exposed to live microbes in general. A vaccine is basically just fake news for the immune system. So you can think of it as a systemic probiotic to educate your innate immune system to more effectively control against septic inflammation. And so that's kind of where I wanted to end it, and I'll let Paul move on to the last slide.
Thanks, Mari. Yeah, that pretty much finishes us up here. We're just want to acknowledge the funding that has occurred thus far relative to the work that's done in the lab and the work that will be done in the next, starting in the next month and progressing over the next year or two. And we're really, really very excited about doing this, and we'll be happy to take questions.
All right, thanks everybody. We have had some questions come in, and I saw that Dr. Navarre was answering some of them while she was talking. But I do want to start from the top. Someone had a comment about no BCG use in Italy and only passed in much of Europe. That catches the eye from a severity correlation standpoint. And that was a comment. I didn't know if you wanted to comment on that.
Well, I think that actually has, it's a great observation too, because they had a real difficult time. And if BCG wasn't being used there in a standard in that country, you can see how those cases could have erupted very quickly.
Okay. Going down, we have some more questions. How is supply chain for MMR vaccine?
Yeah, so that's interesting. In theory, we have lots and lots of MMR available. I have tried to reach out to Merck just to let them know that this, our publication came out and that perhaps they would be getting a lot more requests for the vaccine in adults. They did not respond to me, but I'm hoping that, you know, it's in place. Merck is a very large pharma company. They could boost production fairly quickly for MMR.
Okay.
I also think that they need to be cognizant of this if it's out. I mean, the vaccine's been around for a long time. They know how to manufacture it. They can step up manufacturing to enable to catch the demand and keep the supply up. So I think it's I think it can be done. It just needs some ingenuity.
Okay. Something that's going on in the chat, and y'all, you can post your questions to the chat, please. It should be in the bottom right hand of your screen. But Dr. Navarre is talking about vaccinations or SQ versus IM. Can you just explain to everyone what that means?
Right. So the intramuscular, you're actually, you know, injecting it deep within the muscular tissue, and then a sub-Q is right underneath the epidermis. So that's also different from the way the TB vaccine is administered that ends up causing a lesion. So both routes will lead to systemic uptake of the live organism, will be taken up into the bloodstream and allow it to traffic. In theory, we think it needs to traffic to the bone marrow to educate those stem cells to become these suppressor cells. So both of those routes would work. The only route that I think would not work as well is there was a live flu vaccine available that was the intranasal vaccine. I don't know that uptake through a nasal mucosa would get to the bone marrow as quickly and is at high enough levels to induce this training. So that would be the only one that I would be suspicious that wouldn't work this way.
Okay. And so how far in advance do you recommend people would need this vaccine for the protection?
Right. So based on literature that's out there and based on work from our models, we think that the innate system would be induced within 1 to 2 weeks after getting the vaccine. People have also asked us how long-lived would this innate response last. And in our hands, in mice, it's hard to correlate the mice to humans, but there are also studies in humans with BCG, and they think that it lasts at least a year, perhaps longer. Obviously, with children with these really low rates of COVID symptomatology, it probably in humans might last up to 5 years or more. But it's long-lived, but it's not lifelong. So if you had MMR like I did when I was 5 years old, well, 40 years later, I probably have antibody titers to measles, mumps, and rubella, but I'm not going to have that trained response still available to protect against sepsis.
Okay.
But if we do look at the rates of infection and morbidities, really it starts in the 30s, or some evidence in levels of the late 20s, but teenagers, early 20s, and then your younger children aren't seeing this with high morbidities at all. So it actually could, because of the booster nature of those vaccines and the number of them that children get, because we're saying really any live attenuated vaccine can do this. There could be evidence for these cells well into those teen years.
Okay. Someone is asking, so anyone with good MMR antibody, I think you said titer, physicians won't allow to get MMR vaccine.
Right.
I
had this very same experience. So I requested, I don't have a really long relationship with the current physician that I have. I usually see another physician, but this became my primary care after switching organizations. So I asked to get an MMR, and he requested that I go get serological testing to see if I still had antibodies to measles, mumps, and rubella. And I asked, is that to basically get my insurance to cover this? Will they not cover it if I have positive serology against measles, mumps, and rubella? They said, yes, that was the case. I went to my husband's physician. He just ordered the script. I went to Walgreens and got it, and my insurance covered it without serology. So it varies from physician to physician whether they are going to try to take that next step with serology, but absolutely insurance will cover it if your doctor prescribes it for you. Now, convincing them of the reason why you want it is going to be you know, iffy, because this, again, we haven't tested this scientifically. It's just a hypothesis right now. But the issue is, even if you get the MMR and it doesn't work the way we think it might work against sepsis, there's, again, no harm, no foul. Nothing, you know, you'll get a booster and get better antibodies to measles, mumps, and rubella, which, by the way, are still circulating in this country. So that's my experience.
It is not a problem to get additional antibodies made from a booster if you even have some titers that are present. There's just no harm to it in that sense.
Okay, good to know. All right. Another question that came through, is there anyone who could publish a rigorous study of different countries' experiences covering other factors? For example, American Samoa did really well after their measles outbreak and vaccinations, but they also stopped all ships and planes.
Yes, I mean, there's a number of factors that go into this, and a lot of these countries that are doing very well have done multiple actions to stop the spread and so forth. So it's multifactorial. There's not one thing that's going to do it. I think the ones that really shut everything down and so forth are going to be showing the lowest rates of infection and death rates, as opposed to those that didn't do as much. But again, the evidence is circumstantial, it's anecdotal, but it is positive and supports it.
Mm-hmm. Would you foresee a future where everyone gets a system probiotic yearly? Would this make sense with known pro cons?
Yeah, so I was just trying to type a response there. I think, you know, once we get further along in our clinical and primate studies, and we actually determine how long this trained innate response lasts in humans, whether it's 1 year, whether it's 5 years, I could see a situation where you may recommend a live attenuated vaccine booster, you know, every 5 years or so as a form of systemic probiotic. Again, this would require changing the narrative, not just for the public, but also for healthcare providers, where, you know, this is not taught in immunology classes. I teach immunology for medical and graduate students, and you know, it's so new that it's not even in the textbooks yet, but it should be.
Okay. We have a viewer who's saying that she just went to the local pharmacy and paid out of pocket and asked for it. And it was $110 out of pocket. So that's information for everyone to have there.
Yes, and a lot of local health agencies within your parish, county, city you can walk in and request a booster, and they will administer it very cheaply, if not free. So that's another way to do it that I've been suggesting to people.
Okay, and someone is saying the MMR vaccination is given twice, with the second given 28 days after the first. Do you recommend getting the second booster shot, or is just one sufficient?
Right. So, for example, it's only one and done to induce this trained innate response. However, if you also want the added benefit of a strong antibody response to protect you from measles, mumps, and rubella, you would, in theory, you would need a prime and a boost, so first and second shot. For example, my father, who's 80 years old, never received the MMR as a child because it wasn't available. So he got his MMR for the very first time last month. So for him to get an antibody response, he probably would need a second shot.
Interesting.
Well, we think one is really sufficient for this, based on our work in the models. But I don't think it can hurt either way. But we think that one really induces, and you know, we say they're fairly long-lived, but not lifelong. But we don't have to give multiple doses in our model systems.
Okay. And just so everyone knows, you can see all of a couple people have listed places where they were able to get them either with insurance or just asking over the counter. So if you look in the chat, you can see that. And in the recorded webinar of this, all of the chat will be on the right. So even if you just watch the recording, you'll be able to access this entire chat. So if there are any more questions, please post them now. I'm going to go ahead. I'm going to put up just a little bit of information in the chat for you all, where you can see where you can find more information about the work that these doctors are doing can be found at this page I just posted to the chat. We did have another question. Anything people can do to spread the word about enrollment?
Guess that would be Paul. How are you doing the enrollment for your clinical trial?
Well, initially we are going to the healthcare workers at the University Medical Center. I think we have many able and willing individuals. Our trial at the start is going to be 50 individuals, 25 per arm for the MMR versus placebo. I do plan to submit an R01 grant to the NIH for a clinical trial in October, which would potentially increase that up to 250 with 125 per arm. And hopefully what we do now gets us started for some preliminary data to support that. And you know, I think the grassroots campaign is great, and spread the word that way. And then as we move potentially into nursing homes and nursing home residents, and maybe sports teams get the bug to vaccinate, I think it'll start spreading and continue to spread in a very positive manner while we do the trials and get the data.
Okay. And there is one more question I want to ask. I think this is important. Should kids get another booster before returning to school?
If it were me and I had kids going back to college or school or anywhere, yes, I would definitely do it. Their most recent MMR might have been, you know, if it's a young school-age children, years ago. So again, it won't hurt them unless they have some sort of immune compromise situation. And it, you know, it could impart this benefit. So I would do it. And we've, you know, told our adult children to get it as well.
I would say anything high school and above, high school, college would be very applicable. Grade school, it probably, and,
they may still be
and
protected.
middle school probably still has the childhood effects of this nature. But you know, we'll know more, I think, once we do these trials.
Okay, great. Thank you. And I did post the links to where you can find out more information about the work they're doing, like I said, and also where you'll be able to see the recording of this webinar. Dr. Novair and Dr. Fidel, thank you so much. This was so interesting. We really appreciate your time and your efforts.
Happy to do it.
Yeah,
Yeah, absolutely. I wanted to share one additional web link with you all in the chat. This specific project and many others are part of our coronavirus relief effort. And as Dr. Fidel mentioned, we've partnered with Pharmacies Foundation, and they've provided some funding. But if you go to the link, Give Corona Relief Effort, you can see this project in full detail, along with the webinars and other projects that we are working on to respond to coronavirus. So thank you both very much. It's really exciting, and we are anxious for the data. So we will sure to have you back on with another update, and I look forward to it.
We look forward to that too. Thank you.
All right, take care, everyone.
Take care, everybody.
Bye.